These are ways that genes can be inherited to the next generation, but the way they show up phenotypically does not follow a strict dominant recessive relationship. So there are other ways that genes interact, and these are the four that you're responsible for. So let's start with incomplete dominance. Okay, incomplete dominance means that when you have a blending of the alleles, okay? So let's take a look at the genotypes here. So if a flower inherited both of the dominant CC alleles, they are red. If they inherit both of the small CC, they're homozygous little c, they're white. However, a heterozygous individual, big C, little c, is not red or white, but it's a blending or an incomplete, right? The red is not completely dominant. It's incompletely dominant, so you get a blending, and you get a third phenotype, which is pink. All right, so you could easily do a Punnett square, right? So if you had a red flower, you know that the phenotype is big C, big C. If you had a white flower, it's little c, little c. If you fill in the boxes, then you should be able to pick out which boxes are pink, which boxes are white, and which boxes are red, just by looking at your key here. All right, so that's incomplete dominance. So if we look here, let's take a look at we're crossing two, so let's start at the top. What's the genotype here? Big C, big C. What's the genotype here? Little c, little c. If you created a Punnett square with big C, and little c, then you're going to get 100% pink, because the only gamete that the red flower produces is a big C, so a big C, and then the white little c, so 100% of these offspring will inherit one big C and one little c, which is pink. But in a pink flower, they can create a big C gamete or a little c gamete. So if you cross two pink flowers, big C, little c, big C, little c, then the two big C's would create a red. The two boxes here that show up pink would be heterozygotes, and then the one box would be white. So it's interesting because if you had two pink flowers, the offspring of two pink flowers, you would see 25% of the flowers ending up being, or that's a possibility of having a red flower is 25%, the possibility of having a white flower is 25%, and a 50% chance of inheriting the offspring being pink. All right, so that's incomplete dominance. There's a real-life human example for incomplete dominance called hypercholesterolemia. This is basically a fancy word for high cholesterol. So look at the genotypes down here. If you inherit big H, you have normal cholesterol levels. If you inherit a big H, little h, you have twice the normal cholesterol levels. And if you inherit two little h's, you have 5 times the normal cholesterol level. Okay, so which means that little h is a very, very high cholesterol amount. The big H is normal, and if you have a blending, right, then you have some intermediate cholesterol. So this is actually how this hypercholesterolemia works in humans. So if you have really high cholesterol in the family, that means someone has probably a homozygous little h genotype. Okay, so if one of your parents has normal cholesterol and the other has hypercholesterolemia, what is your cholesterol likely to be? So to work this problem, normal cholesterol, remember that was big H, and then the other parent has hypercholesterolemia, little h, okay? So what is your cholesterol likely to be? So this is a diploid parent. We're going to separate and make gametes, right? So one big H goes here, representing one gamete. They only have big H's to give, so both of their gametes have a big H. The other parent only has little h's to give to their gametes. And if you fill in the boxes, right, you can see that all the possible boxes are going to have a big H in them and a little h in them. So you are as an offspring of your parents, this is the possibility of your cholesterol, right? So 100% possible, right? 100% that your cholesterol is going to be the heterozygous form, which is going to be twice the normal levels. Okay, so if you got that, good for you. Topic is codominance, which means that both alleles are expressed equally in their phenotype. The hybrid in the hybrid in separate distinguishable ways. So what this means is that your both of those alleles are expressed no matter what. One is not dominant to the other, they're both expressed. So blood type is a really good example of this. Let's take a look at your outline. There are 3 alleles for blood type. There's the allele big I with an A on it, big I with a B on it, and little I. So big I with the A on it is going to code for A. So back up. There are, if you didn't know blood types, there's type A, type B, type AB, and type O blood. What these refer to is the kind of glycoprotein or protein with a little sugar group on the surface of your red blood cells. So type A blood only has the A type of glycoprotein. Type B blood only has the B type. If you're AB, you have both A and B, and if you're type O, you have nothing. Okay? So what are the genotypes possible for type A blood? Well, you have to have the A, this big I with the A, which codes for making that A protein, so that you have to have one of those. The other one, though, can be another big A, so this can be your genotype, okay? Or you can inherit, or the next can be big IA and little I. So little I does not code for anything. This is a blank, all right? So because you only have A's, you need to inherit an A, okay? And it can be 1 or 2 A's. For type B, same thing. So if you have to inherit an allele with that creates a B protein on your red blood cells, so you can be a homozygous IB, or you can inherit the B, and the other allele can be that little I. If you're AB, there's only one genotype you can be, because you have to express the A, and you also have to express the B. All right, so this is what that codominant really means, is that both of these alleles are showing up on your red blood cells in equal ways. Not one is dominant over the other, so that is codominance. And then type O, if you have nothing on your red blood cells, then you have inherited 2 little i's, because the little I alleles do not code for anything. Okay, so that is how blood type works. All right. This is the slide which shows you the things that we just worked out on the other page. So this is the A blood type, the B blood type, the AB blood type, and the O blood type. Now let's do this. Mom has AB blood and your dad has type O. What are the possibilities of your blood type? So mom has AB. What is her genotype? Right, she has to have 1 A and 1 B. That's her genotype. What's your dad's genotype? Your dad is type O. So we're going to cross that with little I, okay? So we're going to make a Punnett square, all right? And we're going to put the parents here, big A, B, little I. All right, so in the boxes is all the possible blood types you could get. All right. All right, so hopefully this is getting easy for you. All right, so let's take a look at what we have here. What are the possible blood types you can have? So half of the boxes have big I, big A, little I, so heterozygous for type A, so this is a type A phenotype. Half of your boxes are showing the type B. So you have a 50-50 chance, right? 50% chance of being a type A person, 50% chance of having type B blood type. All right, so that's how you would work that problem. B means that one gene can have many effects, multiple phenotypic traits on the individual. So far, we've talked about how one gene does one thing, right? Eye color, flower color, seed shape. But in actuality, sometimes one gene controls multiple phenotypic traits. On a molecular level, each gene only has one primary effect, but multiple consequences may arise from it through secondary effects. So an example of this is our Devon Rex cat here. So there is one gene that has been altered, one allele, but it affects the hair, the shape of the face, the eye shape, the cheekbones, the ears. So there's many effects that that gene has on the cat. So that's pleiotropy. Another example of pleiotropic effect is Marfan syndrome. Marfan syndrome is basically there's one protein, fibrillin, that is defective. So one of the alleles that you inherit is defective. However, it affects many, many parts of your body, right? The skeleton, the eyes, the cardiovascular system. People with Marfan syndrome have very long limbs and fingers and a concave chest. So this is another example of pleiotropy. All right, and then a topic here is polygenic inheritance. So poly means many, genic is genes, inheritance. So this means that one of your traits, for example, eye color, is actually a combination of many genes. Okay, so that example I gave of how brown eyes are dominant to blue is not real. It's used a lot just to get the concept across of dominant recessive relationships. But in actuality, your eye color is an additive color, and it's based on at least, they think, 3 genes. So your skin color also works this way. So look, actually, let me go back to your eye. So if your eye colors were based on the interaction of 2 genes, A gene and B gene, then you have 16 possibilities for eye color. If you inherit the dominant dominance, homozygous dominance, you're very dark-eyed. If you inherit the homozygous recessive, you're very light-eyed. But actually, there's about 3 genes involved. So let's take a look at skin color, which is 3 genes. So you can see that a 3-gene parent, all the possible outcomes and all the different combinations of those 3 genes have a gradation effect, right? So you can inherit very, very dark skin, you can inherit very, very light skin, or anything in between. So that's why there's so many shades of skin color, because there are just many genes. Your skin color is the additive effect of many genes coming together, not just one. This is another example on height. Height is the same way. You have gene A, B, and C, and you kind of get this bell curve where people who inherit the sort of recessive, homozygous are very short, the dominant are very tall, and then you have all this variation in between. All right, last thing is the role of the environment. This is really that, you know, the eternal question, nature versus nurture. So how much of what your phenotype is based on your genotype, right? Because we know that your genotype is interactional. Your environment can actually dictate how much your genes are expressed, if they are expressed. And so there are many factors that come into whether how much your genotype is expressed into your phenotype. And so this is a very huge question mark in research right now. We don't know how much of your genotype is going to be reflected in your phenotype. You know, if you have the genes that maybe predispose you to cancer, will you get that cancer? Will you have the phenotype? And that largely might depend on your environment. If you have the genotype to be very, very tall, but you're malnourished as a child, you're not going to be tall, right? Your phenotype is going to be altered because you didn't have the environment to really have the genotype be expressed fully. So let's look at the examples I have here. Some genes are really environmental. So what language you speak or the religion you have is predominantly probably what you're exposed to in the environment. Interactional means that there's some interaction between your genes and your environment, like your height, your weight, and skin color, and maybe your health, your mental health, your physical health, things like that. And then predominantly genetic, right? Things that do not change because of your environment whatsoever would be your blood type. No matter what anybody tells you, your blood type is not going to change from any environmental stimulus, and eye color. And, hemoglobin type. You're not going to change the kind of protein you have in your hemoglobin because of your environment. So there are some genes that are predominantly genetic, some that are environmental, and some, this is our big question mark, interactional, right? So scientists are looking into, things like IQ, things like obesity, things like depression and mental illness. How much of this is interactional? This is an example of clones. So this represents the same exact genetic clipping from a plant. So they planted this plant at different elevations on a mountain, and they found that even though genetically the plant is identical, that the way it grew at low elevation, medium elevation, and high elevation are drastically different. So it looks like different plants. They behave differently, they grow differently based on their environment. So this shows you that interactional relationship with genetics and the environment. Okay, so again, nature versus nurture, big question in science, right? How much of our physical being is nature and how much is nurture? All right, so that's it for this one.